Yes. Okay, we're ready. We are recording. So, welcome everyone to this new cycle of seminars from our institute. Uh, well, most of you were already here last year, so you know that we started last year with these webinars, doing them online due to COVID. And this year we decided to continue the same way. So we are going to have all virtual seminars this cycle because, well, most of you said that you prefer that. And also because taking advantage of this situation, we decided to invite some experts that are working abroad. And for that reason, some of our seminars will be in English. Okay, and that's the case of today's seminar that will be in English. Most of the other ones will be in Spanish, but we will have like 3 or 4 seminars in English. Okay, just because this is the first seminar, I just want to do a quick reminder for those of you that need a certificate of attendance, that please, it's very important that you log in to the seminars with your full name, okay? We won't count your attendance if you log in just with your name or your nickname or with initials, okay? So no exceptions with that. So please, those who need the certificate of attendance, just log in with full name. And well, I think that without further delay, we can start with the first seminar. So it's my big pleasure to introduce you our speaker today. She's Professor Darlene Dartt from the Schepens Research Institute. Most of you already know her, and those who don't, you probably should. Okay. Okay, because she is one of the world leading experts in ocular surface physiology. Just a very brief introduction. She's full professor of ophthalmology at Harvard Medical School. She gets her PhD in physiology at the University of Pennsylvania. After that, she moves to Europe. She came to Europe for a postdoc in University of Copenhagen in Denmark. Then she was postdoctoral researcher at Tufts University in Boston. And after that, she started Harvard Medical School, where she has developed most of her career. She's working at the Schepens Eye Research Institute, as I said, being a senior scientist there, and she studies the neural regulation of tear production. And specifically, she focuses on signaling pathways that nerves and growth factors use to stimulate protein secretion from lacrimal gland and also mucin secretion for goblet cells. She's really one of the greatest experts in conjunctival goblet cells, and today she's talking us about this topic and how is the regulation of this conjunctival goblet cell function in dry eye disease. So thank you so much, Darlene, for accepting this invitation. It's an honor for us to have you here. So whenever you're ready, you can start.
Okay. Well, it's a real pleasure for me to be speaking to you all at Ioba today. I've had a long association with Ioba and really enjoyed my scientific and social interactions with many of you there. And today I miss seeing you all and interacting, but I also miss the delicious food that I always get when I come to visit via Dartt. So now with the science. So today, as Laura mentioned, I'm going to be talking about the role of nerves and cytokines in regulating goblet cell function in dry eye disease. So I would first like to acknowledge the people who have, these are people who've worked in my lab and the ones who specifically worked on the project. First was my longtime lab manager, Robin Hodges, and then the many students in my lab on this project: Sumit Bhattacharya, Yukako Tadamatsu, Kalbi Khan, Tomas Blanco, who I think many of you know, Laura Garcia Posadas, who you know, And then my longtime collaborator, Sharmila Masli from Boston University School of Medicine, and Laura Contreras-Ruiz, who I think many of you know, worked on the projects with Sharmila that I'm going to include in my seminar. So I'd like to start by just a short overview. So the anterior eye functions to protect the eye, the entire eye, and the ocular surface from the external environment. So there's a lot of challenges and stresses in the environment, including mechanical ones, thermal, chemical, pathogenic, desiccating stress, and pollutants, which is becoming ever more important. But the eye is really well equipped to deal with all these stresses and challenges. There are the lids that close quickly with the blink. There's the tear film that in particular contains a lot of antibiotic type proteins, antibacterial, the cornea, and the conjunctiva. So the cornea helps protect the eye because it has such a tight junctions in the epithelia. However, the cornea has one sort of Achilles heel in that it has to maintain transparency. So it's limited in how it can respond to the environment. But the conjunctiva is not transparent. It's a very leaky epithelium, has a lot of blood vessels. So it's able to respond to the environment and has really a multitude of redundant mechanisms for this response. So first are the epithelial cells of the conjunctiva, the goblet cells, and the stratified squamous cells. There's also the loose stroma of the conjunctiva. But today I'm going to talk about the goblet cells, and these cells are particularly important because they synthesize and secrete this high molecular weight mucin, MUC5AC, that is particularly protective for the ocular surface. So this slide shows the tear secreting epithelia. So here's a schematic of the eye, and this green layer here is the tear film. And the outer layer is secreted by the meibomian glands in the lids, the aqueous layer by the lacrimal gland, and finally the mucus layer or the aqueous mucin layer. It's sort of unclear if there's 2 layers or 3, so I'm going to hedge on that today. So the goblet cells are interspersed throughout the conjunctiva, upper and lower lids, and this is an electron micrograph of them. This is the stratified squamous cells that surround these goblet cells, and they're full of these secretory granules that you can see here, and these contain MUC5AC and other proteins that the goblet cells have synthesized, and they're released into the tear film of the ocular surface. So today I'm going to talk about a lot of what regulates this release. So, and our sort of basic premise is that goblet cell secretion and number is regulated in health and disease. So this conjunctiva here represents homeostasis or the physiological state. And here are the goblet cells and the stratified squamous cells, and there's a certain optimal number of goblet cells, and this represents the MUC5AC and components that the goblet cells secrete. So under health, you need a certain amount of goblet cells that secrete a certain amount of mucins. So in ocular allergic disease, which we've studied, but I'm not going to talk about today, there can be more goblet cells, and it leads to more mucin production and overproduction of mucin, which is deleterious to the surface of the eye. And in contrast, in dry eye, there usually are fewer goblet cells and less mucin. So this, because these are 3 different states, this suggests that the mucin secretion is or the goblet cells and their function are highly regulated. And the regulation comes from MUC5AC synthesis and secretion, and goblet cell proliferation and apoptosis. So before you can really understand disease processes, it's important to understand how goblet cell function is regulated in ocular surface health. So it's well known that nerves stimulate lacrimal gland secretion of electrolytes and water. And in this schematic here, there are sensory nerves in the cornea and conjunctiva that send impulses to the central nervous system in really a complex layers of reflexes. So this is the first part. And then they come out to activate the efferent nerves, predominantly the parasympathetic nerves in the lacrimal gland, but also sympathetic. These nerves release neurotransmitters that activate specific processes that cause lacrimal gland fluid secretion onto the eye. So we believe that goblet cell secretion was regulated in a similar way with the afferent nerves, efferent nerves, and stimulation of signaling pathways in the gland. So this was not what was considered to be how goblet cell secretion was regulated when we started this project. They were known as basal secretors. So here's a bigger picture of the EM of the goblet cells and the stratified squamous cells. So we've used predominantly rats and human tissue for our studies. And Sharmila Mosley has used mice, and they all are pretty much the same, actually. So this is a rat conjunctiva, and we wanted to see if nerves stimulate goblet cell secretion. So we first looked to see if nerves were present in the epithelium, and I'm not going to show you any pictures for time, but they are present, both parasympathetic and sympathetic, and their endings come in around about here. So this is the basal layer of the gland of the cells. So it's not at the bottom where we thought, but up around here. And then we looked for muscarinic receptors because acetylcholine is released by the parasympathetic nerves, and they were up in this area as well. So the nerves and its receptors were perfectly poised for nerves to stimulate goblet cell secretion. So we've done experiments in several different types of preparations. So the first we looked at was the rat in vivo, and in one animal anesthetized, we did nothing to the eye. And the second animal, we made a corneal wound, and that was our way of activating the sensory nerves. And then we took out pieces of tissue from the same place. And we stained them with Alcyon blue PAS, and that stains the secretory granules in the goblet cells. So you can count the number of goblet cells in this area by counting the number of cells, which you can see are delineated here per unit area. So these are cells that contain their secretory granules, so they have not secreted. After a corneal wound, you can see that lots of cells have lost their secretory granules, they've secreted them, and you can count the number of cells that are left. So in an EM, this would be goblet cells that haven't released secretory granules. You can see they contain pretty much all of them. And here are the ones that have secreted. So you can see when goblet cells secrete, they release all their granules, and that's why this assay works. These are secreted and these are not. So we graphed this and we set all the controls to 100%, and we did experiments at different times, and you can see at time dependence secretion of goblet cell mucin. And so a decrease is an increase in secretion. So this shows in an in vivo situation that the cells are stimulating the goblet cells to secrete. In that situation, you really can't do too many experiments in too many conditions in a single animal. So we then moved to conjunctival pieces, and we looked at the muscarinic receptors. So here we're looking at secretion, and we're able to know this is goblet cell secretion because of the MUC5AC secretion. And this is control over time, and this is the cholinergic agonist carbachol, stimulating secretion in a time-dependent manner and in a concentration-dependent manner. We then looked at the type of muscarinic receptors that were used. So this is carbachol that activates them all. Atropine blocks them all, so that decreased secretion. And this is a mainly M1, M2, and M3 receptors, and each of them significantly decrease secretion. So all of the receptors, M1, M2, and M3, play a role in the parasympathetic stimulation of secretion, as does VIP, which is the other parasympathetic neurotransmitter. So this type of preparation gained us additional information, although it proved very difficult to work with. We also wanted to find out information about the biochemistry about the goblet cells, and because the pieces in vivo contain both stratified squamous and goblet cells, you couldn't really do any biochemistry except secretion. So Marie Chatos in my lab then developed a cell culture model where we could culture primary and first passage goblet cells, and we've characterized them quite extensively, as you can see here. And you can see our cultures are really overwhelmingly goblet cell. So using the goblet cell preparation, we repeated the secretion by the cholinergic agonist, and again, we get a time-dependent stimulation of secretion. So we've done a lot of work using the cultured goblet cells, and we came up with this conclusion that similarly to the lacrimal gland, stimuli from the external environment interact with the afferent nerves of the cornea and activate them. And then through this complex reflex, I keep gesturing with my hand instead of my pointer, activates the parasympathetic nerves that surround the goblet cells, and they release their receptor neurotransmitters and stimulate secretion. We also looked a lot about the cellular signaling pathways and have done that in even more detail than shown here if anyone is interested. But this shows that muscarinic receptors, VIP receptors, EGF receptors, for a couple of them, when they're activated, they increase calcium and the P44, P42 MAP kinase to stimulate secretion. So we feel we have gathered enough evidence to show that, like the lacrimal gland, the goblet cell secretion is regulated by nerves. And that makes a lot of sense because you'd want the goblet cells to really respond very quickly to the external environment. So now that we've looked at the normal regulation of goblet cells, we can then look at to determine or ask if they have a role in pathogenesis of dry eye disease. So as most of you know, dry eye affects over 40 million Americans and a lot more individuals, obviously, around the world, and that limited treatments are available, although some new ones have been developed over the years. It is a complex multifactorial disease where tears are not properly produced, not of normal composition, or evaporate too quickly. It's usually accompanied by ocular surface inflammation, damage, and pain. And what we're going to look at is decreased corneal and conjunctival sensory nerve stimulation can play a role, and dysregulated mucin secretion can also contribute. So this is from the DEWS 2 workshop, and it sort of is the consensus pathophysiology of dry eye. And you can see there's aqueous deficiency dry eye and evaporative dry eye. Evaporative dry eye is due to meibomian gland function, and aqueous deficiency to lacrimal gland dysfunction. There's really no one type of goblet cell dry eye, but you can see goblet cell and goblet cell dysfunction is right in the middle of this vicious circle that causes the tear, hyperosmolarity, the evaporation, and the signs and symptoms of dry eye disease. So we're going to look at a couple steps in this pathway to see if they play a role in regulating goblet cells or dysregulation of goblet cells. So there are two types of aqueous deficiency, non-Sjogren's and Sjogren's, which is autoimmune. So we're going to use a mouse with autoimmune dry eye, which is the thrombospondin knockout mice. And we're going to look at the reflex blockade, so the role of nerves, and then the role of cytokines. So we're going to use a thrombospondin knockout mouse, and thrombospondin is called the master regulator of cellular signaling, and its different components are shown here. So it can function both extracellularly and intracellularly. It's a scaffold protein for multi-protein complexes. It has multiple signaling domains, as you can see here. And these signaling domains 1 are what activate TGF-beta. And in the lacrimal gland and goblet cells, activation of TGF-beta causes an anti-inflammatory environment. It has these type 2 domains that are EGF-like, which gives the potential for EGF mimicry or inhibition of EGF-like responses. And then type 3 domains, which bind calcium and can have a potential effect on calcium. But we're using the TSP for its activation of TGF-beta. So Sharmila Masli developed this mouse model and described it. And so in this model, dry eye develops spontaneously with age. So this is lacrimal gland from wild type and TSP knockout mice, increasing age. And you can see here in the TSP null mouse, this is, I think, about 24 weeks. So this is 8, 12, 24, 48 weeks. So there starts to be some inflammation and invasion of immune cells around 24 weeks. In females, it occurs a little bit earlier. And by 48 weeks, there are these large lymphocytic foci in the disease gland, but only very little cells in the wild type. And you can see this increase in foci. And then here you can see with age, sometimes the eye gets completely destroyed. Okay, so in the lacrimal gland, the dry eye develops spontaneously with age, unlike other organs in the body, which need something to induce a disease to look at the effect of TSP. And in dry eye, activation of TSP improves the disease because TGF-beta is anti-inflammatory. In other places, it's the block of TSP that improves the disease. So why is the ocular surface different? And the reason is that there's mainly TGF-beta type 2 in the ocular surface, where it's type 1 in other places. And type 2 TGF-beta is only activated by TSP1, whereas type 1 is activated by integrins as well as TPA, as well as thrombospondin. So we're going to look in this mouse model at what's happened to the afferent pathway, the efferent pathway, stimulation by exogenous neurotransmitters, and stimulation by cytokines. And this is back from our model of the goblet cell. So the afferent pathway, efferent pathway, stimulation by exogenous neurotransmitters, and cytokines. So we looked first at the cornea, and we determined when cytokines were increased in this tissue, and this is wild type and TSP knockout at 4 weeks, wild type and TSP knockout at 12 weeks. And this is when you can see some disease, and this is little to no disease. And you can see that the cytokines MCP1, MIP2, and TNF-alpha were all increased in the TSP cornea around 12 weeks of age. We then looked at the nerves. We looked both in vivo by confocal microscopy, and this is by a staining of fixed corneas. And we used that antibody TUJ1, which recognizes all types of nerves. This is wild type at 4 weeks, TSP at 4 weeks. You can see a small decrease even at 4 weeks. This is wild type at 12 weeks, and can see there's a really significant decrease in nerves at 12 weeks in the TSP mice. And we quantitated that, and there was a small but not significant decrease at 4 weeks, but a significant decrease at 12 weeks of age. And the CGRP, we used specific antibodies, and the CGRP containing nerves were the ones that were decreased, whereas the substance P ones were not altered, which is interesting because CGRP tends to be anti-inflammatory, whereas substance P tends to be pro-inflammatory. So we were able to show that there was a decrease in corneal nerves, but we did not do any experiments to determine if the nerves were dysfunctioning, although looking at the changes, you would suspect that they were. So it looks like that with the afferent corneal nerves, they're decreased and that's a direction that would cause dry eye. So next, we're going to look at the efferent stimulation. So we looked at the parasympathetic nerves. That's the predominant innervation of the lacrimal gland. Top panels are wild type, bottom panels are TSP knockout. And so these are the TUJ1 staining the nerves, and you can see that they really surround the lacrimal gland acini. This is at 12 weeks when they're diseased. We did a whole series of time dependency, but I'm just showing this one. And you can see at 12 weeks when there is disease that you've really lost a lot of nerves. Floydin outlines the goblet cell membranes, and you can, then when we merge it, you can see the nerves are green. There's a lot of green here, but really a lot is lost in the mice. So like the afferent nerves, the efferent nerves are decreased too. In the lacrimal gland, we can look at the function of these nerves. We can take pieces of lacrimal gland and stimulate them with high KCl, which causes the nerves to release their neurotransmitters. So we did this at 4 weeks, 12 weeks, and 24 weeks. And the dark panels are wild type. The light ones are TSP. So this is basal secretion, and this is high KCl. And in the normal mice, there's an increase, although not significant, at 4 weeks, and a decrease, again, not significant. But at the remaining times, secretion is impaired in the TSP knockout mice. So this suggests for efferent stimulation that parasympathetic nerves are decreased and their function is decreased. And since the nerves in the goblet cells in the conjunctiva come from similar pathways as the lacrimal glands, and 2 types of nerves were decreased in the knockout mice, we feel confident that the nerves were decreased in the goblet cells as well, and their function was most likely impaired. So that would lead in the direction of dry eye. We're next going to look at neurotransmitter stimulation. And now we're returning to the goblet cells. And this is work done by Laura Contreras-Ruiz in Sharmila Masli's lab. And they looked at the conjunctiva of TSP knockout mice, and they're looking at 6 weeks and 12 weeks, wild type and TSP knockout. This is the conjunctiva, both upper, that surrounds the either upper or lower. You can see it here, and these little white spaces, clear or lighter spaces, are the goblet cells in this H&E section. And at 12 weeks, you can see that there's a lot of inflammatory infiltrates in the TSP, but not the wild type and not in the younger mice. So similar to the lacrimal gland with aging, you're getting infiltration of the conjunctiva. Then Sharmila and Laura looked at MUC5AC in tears, and it was tears stimulated by cholinergic agonist. So both wild type and TSP knockout basal were similar. And as the disease progressed, there was a significant decrease in the amount of MUC5AC in tears. So this is, I can't see it, so like 6, 8, and 12 weeks. They also looked at MUC5AC expression in the cornea, and that was decreased with age as well, not at the earlier times. So it looks like there's infiltration, and along with that, a decrease in MUC5A secretion, expression and secretion in the goblet cells. And then they used cultured mouse goblet cells from both wild type and TSP knockout. And these are wild type, and there was no stimulation or stimulation with the cholinergic agonist carbachol in wild type and TSP knockout mice. So at 4 weeks, carbachol stimulated secretion in both wild type and TSP null. But at 22 weeks, in this case, it stimulated it wild type, but TSP knockout mice were not stimulated. So this is a second method showing that secretion was altered with disease development in the TSP mouse. So again, neurotransmitter stimulation of secretion is decreased in these knockout mice consistent with dry eye. So now we're going to look at cytokine stimulation. And again, this is work done by Sharmila and Laura Contreras-Ruiz. And they first looked at different types of cytokines in the conjunctiva of TSP knockout mice. So wild type TSP, 6, 8, and 12 weeks. So TNF alpha was increased in the TSP knockout mice, as was interferon gamma. These are the Th1 cytokines. Th17 cytokines were increased as well, and not shown here, Th2 cytokines were all increased. So a lot of the cytokines were increased with increasing disease. So which ones are active at blocking function? So this is the given cytokine here, and these are just in regular mouse goblet cells, carbachol, and then the cytokine given before the carbachol. And interferon gamma decreases carbachol or cholinergic agonist secretion, as does TNF alpha, so the Th1 cytokines. But the Th17 cytokines do not, so this is increased and this is not significant. And the Th2 cytokine, IL-13, doesn't either. So even though all these cytokines are increased, only the Th1 cytokines, interferon gamma and TNF alpha, are blocking secretion. And if you remember, these were the ones that were playing a role in that vicious circle that I showed you. So in my lab, Laura Garcia Posadas did some experiments looking at the Th1 cytokine interferon gamma, and we looked in rat and human goblet cells. So she first looked at proliferation. So this is basal set at 1, and interferon gamma by itself inhibited proliferation. Carbachol by itself didn't alter it, and interferon gamma didn't alter the carbachol response. So interferon gamma is decreasing goblet cell proliferation. It's a small decrease, but over time could really add up to a large change in cells. So then we looked at secretion of rat and human goblet cells. So this is a 15-minute pretreatment, which didn't work. So here is when we gave a 24-hour treatment. So this is basal. This is interferon gamma by itself. This is carbachol, and interferon gamma 24 hours before the cholinergic agonist blocked it. And the same with human. This is 15 hours, and this is these are 15 hours given after carbachol here, and 15 minutes didn't block it, but 24 hours did. So this is further confirmation that the Th1 cytokines are decreasing secretion and also decreasing proliferation. So to just summarize the effect of the cytokines, the Th1 cytokines are blocking mucin secretion. The other ones are not. The Th1 cytokine is blocking proliferation. The others, these stimulated, in fact. And the Th1 cytokines are causing apoptosis, all which would lead to dry eye. So if we look at our summary here, there are multiple ways which dry eye can be caused in these autoimmune diseases. First of all, decreased nerves and afferent stimulation, decreased parasympathetic nerves and their release of neurotransmitters, decreased neurotransmitter stimulation of synthesis and secretion, and only Th1 is decreasing decreasing multiple functions of the goblet cells. So if we go back to our original diagram here, the cornea activates the efferent nerves, and in dry eye, a loss of efferent nerves can be leading to dry eye via the goblet cells. A loss in parasympathetic nerves can be causing the dry eye. A decrease in neural agonist stimulation of secretion and an increase in Th1 cytokines can also be leading to a decrease in secretion. And we haven't yet explored the cellular signaling pathways that these would affect in the goblet cells. So to summarize, to conclude, actually, neural regulation of goblet cell secretion of MUC5AC maintains and protects the ocular surface in health. Damaged nerves, both efferent and afferent, and increased Th1 cytokine production, decreased goblet cell proliferation, and MUC5AC synthesis and secretion, contributing to the pathophysiology of dry eye. And using these mouse models and exploring how goblet cell secretion is regulated on a cellular level, I think can provide new ways to develop treatments for dry eye. Thank you very much, and I'd be happy to take questions.
Thank you. Excuse me. Thank you very much for this seminar, Darlene. So if anyone can ask a question, you can just turn your microphones on and just ask. Okay. Marga Canonge.
Darlene, hi.
Hi, Marga.
Very nice presentation. Very nice to see you.
Yes, excuse me a moment, Marga. Darlene, if you want to see everyone, you can just stop sharing the presentation in the presentation mode. Then you will be able to see everybody. Sorry, because we have a problem and she wasn't able to see us while she was sharing.
Okay, there we go. So, and then is there a gallery? Because I see you and Margaret now.
Yeah, probably the rest of the people just have.
They have the video off, I would think.
Yeah. Oh, okay. All right.
Okay.
Amalia's there.
Oh, hi, Amalia.
Go ahead, sorry.
So my question to you, Darlene, is if I understood well, a decreasing in afferent nerves will cause a decreasing goblet cell function, right?
Yes.
Right. So translate that into the cornea. We know that in dry eye there is also a decreasing nerve density, and that will explain the same thing. But what about diseases such as atopic keratoconjunctivitis, where there is also a decreasing corneal nerve density, but goblet cell function is increased? How do you explain that?
So we explain that because there's other things in addition to nerves that regulate goblet cell secretion. So we've found that histamine released from the mast cells during allergy can stimulate goblet cell secretion. And there are 4 histamine receptors. They're all present in the goblet cells. So there are other components in addition to nerves that are regulating secretion.
So that will explain also, because I've been told many, many years ago, actually when I was in Boston, so many years ago, that any chronic inflammatory ocular surface disease eventually produces or eliminates or decreases the amount of goblet cells. Would that be the reason? Because even if it is a atopic disease, if it is chronic, goblet cells will diminish. So that might be the explanation, right? That the nerves, the afferent nerves diminish.
Yeah, because actually that's very different from the seasonal allergic conjunctivitis, and it's more long lasting. So you can get. So we do a lot of study of inflammation. So you could get acute inflammation, but then when the disease doesn't, when that isn't resolved, then you can get chronic inflammation, and that can cause a lot of damage to the tissue. So I think the inflammatory process could explain why you're getting a lot of damage. And in the and there's also effects of a lot of the inflammatory mediators on nerves as well in terms of causing pain. So it's obviously a lot more complicated than I presented today in terms of dry eye.
Can I ask one more?
Yeah.
That links with a therapeutic question. Secretagogues, drugs that are supposed to increase the amount of mucus secreted by goblet cells. I really do not believe in these drugs, because I think a goblet cell that is diseased, that is sick, will not increase the amount of anything, but that you need to actually get rid of inflammation so that these cells will start function normally. And that's our experience in clinical speaking, although we don't have a lot of drugs to use, but they usually fail. Would you like to comment on that?
So are you talking about ones like ribamapide and
yes, or
INS type of ones? Yeah.
And another one that is in Japan that I can't remember right now, but topical or even not.
I think that's the ribamapide. It has a different name now, though, I think.
Yep.
I think. So, I mean, obviously diseases, I'm not going to give a really good answer. Diseases are very complicated. And I think that they have to be, studied. That disease needs a model, or you have to study it in humans, that you need to really follow the trajectory of the disease and try to dissect out, are the nerves affecting it? Are the afferent nerves? Are the efferent nerves? What kind of milieu are the goblet cells in? How much MUC5AC is in the tears? How much is left in the conjunctiva? And really get a very good idea of the pathogenesis of the disease before we could start treating it.
Diquafasol was the name that I couldn't think of. Diquafasol.
Yeah, that's the P2Y2 antagonist.
Mm-hmm.
Yeah, and also, it's a multifactorial disease. So, some of these drugs treat maybe one thing, and as you said, the tissue is still diseased, so they're only a treatment, and they're not really repairing the
Exactly
disease,
my point. Yeah.
and they're only treating like one component of the disease. And it's really, there's not even one good metric to tell that you have dry eye disease.
Yep. Mm-hmm.
So, that it starts being, are you sure they have dry eye disease? What kind of dry eye disease do they have for dry eye? I think allergy is probably a little easier.
Mm-hmm. All right. Thank you very much.
Sure.
Thank you. Is there any other people who wants to ask?
Can you hear me?
Yes, I can. Oh, Mike Stern.
Sure.
Hi.
Darlene, your usual spectacular presentation. I just loved it. And you're totally international this morning.
Yeah, how exciting.
Welcome from California.
It's pretty
I
amazing,
do have a question
isn't it?
for you. So some work actually that came out of Ioba originally was the role of EGF and EGF receptor, and it was the one thing, while all the other inflammatory cytokines were going up, it's the one thing that went down, and it was on your slides. You mentioned it there. I was just wondering if mechanistically, if you might comment on that. Why is that part of the loss of protection of the ocular surface, or what's the role of that?
Yeah, so EGF is very important for epithelia, and the goblet cells are epithelial cells. So we actually have done a lot of work on EGF and goblet cells. So it does stimulate goblet cell proliferation, and it also stimulates goblet cell secretion. So it stimulates it on its own. So if it's and so EGF is part of membranes, and it's released by ectodomain shedding, and then it can interact with its own receptor. So it can itself stimulate secretion, which it does. And then we found that cholinergic agonists also will stimulate that EGF ectodomain shedding and cause secretion. So they work through EGF as well as other normal pathways. So EGF, and I think histamine works through EGF as well as other pathways. So there's EGF is an important role in the goblet cells.
Again, really great presentation. Not much could get me into a presentation this early in the morning, but it was really, really wonderful. Thank you.
Thank you very much. It's great to see you.
Thank you. Thank you very much. Anyone else want to ask? Don't see any hand. Yeah, well, what I can say is when I was in the lab working with the goblet cells and doing all these cell signaling studies with the calcium and all like that, I remember that at the very beginning, like goblet cells react to almost everything I was trying. And I say, okay, I'm doing something wrong, let's try. And then we check with different controls, and I mean, things were fine, they were not reacting. It's just that I think they are really very, very complex cells that have multiple receptors to almost everything, let's say. So they react to so many cholinergic receptors, adrenergic receptor, muscarinic, I mean, everything. So they are really very, very complex cells that are able to modify a lot of things. And also all these, that's the studies that Laura Contreras-Reed was also doing about this crosstalk communication between goblet cells and dendritic cells and things like that. So I think that, I mean, they are very much more complex than what most people know.
Yeah. No, it's really, as Laura said, fascinating that, I mean, if you look for histamine receptors, there are 4, and the goblet cells have all 4. I think they're almost the only tissue that has all 4 of the receptors. And I think my idea is just that they have a very important function to protect vision, and the cornea is constrained, although, it does have multiple ways of doing that, but the goblet cells are not. So they just have a lot of redundancy to secrete the mucins, and they also secrete electrolytes in water to protect the cornea.
Yeah, and I think that's what Marga was saying about the secretagogues. I think that, I mean, of course, I don't have any idea from a clinical point of view, but I mean, this is like a simple approach to think like the goblet cell is just something that is secreting. That's all.
Yeah.
And it's not. Probably that's why they don't work, because if the goblet cell is not healthy enough, it doesn't matter if it secretes the content. Is that the content is not going to be the appropriate one. The connections with the immune cells is not going to be the right one. So probably that's why they don't work.
Yeah. Yeah. So so the conjunctiva and goblet cells has really been a great area of work, because everyone concentrates on the cornea and they think that's the important part, and it is important, and they all forget about the conjunctiva. And the conjunctiva is really a unique tissue, and everything's there. So anything you want to study, basically, you can find in the conjunctiva.
Yeah, that's true. Yeah. Okay. So don't be shy and just take this opportunity to ask Professor Dartt about any question you have.
You'll probably ask me something I can't answer. That's what usually happens.
No. Okay, so I think we don't have any other question. So, well, it's just thank you so much, Darlene, for this fantastic seminar. We really enjoyed it, and it's always a pleasure to see you again. And yeah, I hope next time maybe with you can visit us for this good food and better wine that we have here.
And the wine, and then what are those almond cakes that I like so much, with all the nuts in them? Turron.
The turron.
Turron.
Yeah, that's only for Christmas, but
I like them anytime. I didn't know that, so
well, usually
I always have a wonderful time when I come to visit.
Yeah, so you're always welcome. Okay, so I think that's all for today. So thank you everyone for being here today in this first seminar. I think it has been a great success. We have more than 70 people, which is not bad at all. So thank you so much, Darlene, and see you all in a couple of weeks with a new seminar. That one will be in Spanish, and Doctora Esther Carreño will be talking to us. So I will send you the information as usual next week. So good afternoon, everyone. Well, good morning, Darlene. Good morning, Mike.
Yeah.
Enjoy your day.
Thank you very much.
Bye-bye. Thank you so much.
Bye-bye. It was a pleasure.